Hello, my name is Gabriella Schroeder, and today I'm going to be comparing the two gut microbiomes of two different species. And so before I begin, there's some background information I want to go over. First, a microbiome is defined as the types and or amounts of microorganisms present in a particular environment. And so in this study, we're specifically interested in the gut microbiome. And the reason we're interested in the gut microbiome in particular is because it has shown correlation between the gut microbiome and other important physiological conditions. So when we're referring to the gut microbiome in relationship to other important physiological conditions, what we mean is that there's been studies conducted to test the relationship or correlation between the gut microbiome and other diseases such as Alzheimer's, Crohn's disease, irritable bowel syndrome, as well as depression and autism. So with some of our background research, we revealed a study in which a neuroscientist, Dr. Paul Patterson from California Institute of Technology, conducted research in which he compared the fetuses from pregnant rats and whether they would have autism depending upon the presence of molecule known as the maternal immune activation module. And he was trying to test whether the presence of this MIA molecule would cause autism versus like the ones without it. And so this study revealed that the mice with an abundance of the MIA molecule or module molecule had abnormal microbiomes, and specifically they had a difference in the presence of bacterial classes of Clostridia and I believe Bacteroideae. And so this difference was like it was more abundant in the MIA mice than the mice with normal. So MIA mice were the ones who typically had a correlation to autism, fetuses with autism. And so we kind of concluded with this that imbalances with the microbiome may not be the same as humans, but autistic behavior may be rooted in the gut and not the brain. Another study that our background research found was a doctor for the Catholic University of Leuven in Belgium, Dr. Reyes, conducted a study in which she was comparing the microbiome of Belgians and those who had depression and those who didn't. So in this study, a total of 1,050 Belgians were surveyed and 173 were found to have depression. And of those 173 Belgians who had depression, they were able to prove that there was two kinds of microbes that were missing from their microbiome. And this would include the Coprococcus and the Dialister. And the findings were held true when age and sex were taken into account. So moving on specifically to our study, our question was, will there be a difference in gut microbiomes between a snake and a bearded dragon? And our hypothesis was that we believed that a difference would occur between the gut microbiomes of the two different species. And our rationale behind this was, if these two species eat differently and live in different environments, then the bacteria in their gut should be more accustomed to their diet, specifically from what they're eating from their environment. So kicking off with our methods, I want to introduce our two species. Our first was a corn snake. He was aged at 12 years old, and he was a male, and his diet reported by his owner was he ate mice. And the captive lifespan of a corn snake can be approximately 15 years. Our other species would be a bearded dragon. His age was not listed, but we do know he was a male, and the diet reported by the owner was that he was a vegetarian, and his captive lifespan was from 8 to 12 years. So it's important to understand some major differences and similarities between these two species. First of all, some similarities are they're both reptiles. They both obviously have scales, and they are oviparous, and they shed skin, and they are ectothermic. For the corn snake, it is a carnivore. They naturally occur in North America, and they are native to wooded environments. On the other side, there is the bearded dragon. Bearded dragons are omnivores. They are native to Australia, and they're native to deserts and semi-arid woodlands. So the process of this experiment included 3 major steps for analyzing the gut microbiomes of the 2 species. And specifics, our first step included isolation of the DNA from fecal scat samples. From there, we had to amplify the DNA found in the samples using a PCR, otherwise known as the polymerase chain reaction. And then our third step included analyzing the nucleotide bases or sequences using a sequencer. So, kicking off with more details, this first specific step of DNA isolation, we used a kit called QIAGEN, and it was manufactured by QIAGEN Incorporation. The purpose of this step is to isolate the DNA using a series of washes to remove impurities from the fecal sample, so that way we are only focusing on DNA molecules. And the DNA isolation was conducted according to the QIAGEN protocol. Our next step was to then move on with DNA concentration. So now that we've isolated DNA using the QIAGEN kit, we're now moving on to use the Zymo kit, which is again manufactured by Zymo Incorporation. And the purpose of this is to increase the DNA that we had previously isolated by creating a concentration that was tenfold of our isolated product. And this is that we can use more for the experiment. The isolation was conducted according to the Zymo Incorporation protocol, and no observed changes were made. The next step was to quantify the samples, and in order to do so, we used the device called the NanoDrop 2000. And the purpose of quantifying these samples is to determine the concentration and purity of the DNA samples that we have now isolated as well as concentrated. And in order to do this, we had to understand that DNA absorbs ultraviolet light at 260 nanometers, while protein will absorb at 280 nanometers. And this is important in establishing the ratio that we use to determine the purity of the sample. And the purity of the sample was determined by a 260 and 280 absorbance ratio. And the samples that had a ratio of about 1.5 to 2.2 were considered pure, but samples below 1.5 were considered to be contaminated with protein, while samples that had an absorbance above 2.2 were considered to be contaminated with RNA. So the next step was then to conduct a polymerase chain reaction, and this was done in order to amplify or make copies of our 16S RNA gene. And the importance of the 16S RNA gene, or the reason why we're amplifying it, is because it contains 2 highly variable regions known as the V3 and V4 region. And these 2 regions are really important in helping us determining and classifying the bacteria present in the gut, and that are specific to our species. So the steps when conducting a polymerase chain reaction occurs first as a process at a first hold, where we denature the DNA at 95 degrees Celsius for 3 minutes. This is when the DNA strands will separate and all the base pairs are exposed. And so from there we conducted 25 cycles of denaturing at 95 degrees Celsius for 30 seconds, and then we added or annealed primers at 55 degrees Celsius for 30 seconds, and then DNA synthesis occurred at 72 degrees for 30 seconds. So all these steps were done consecutively for each cycle, and there was 25 cycles. Then we continued with our second hold, and it was held at 72 degrees Celsius for 5 minutes for the final extension of the molecule, and then it was a third hold at 4 degrees Celsius where we were able to store our PCR products. From there, we wanted to determine if our PCR was accurate. Did it work? Did it produce enough DNA molecules, basically for us to sequence. And in order to do this, we then conducted a gel electrophoresis. And a gel electrophoresis is important because, as you can see in the image right here, that we put our DNA samples, our PCR product samples, into these wells, and then we run them from an anode to a cathode, so from a negative charge to a positive charge. And the reason we're able to do this is because DNA molecules have a negative charge, so they will run through the wells to the positive charge. Fragments will separate according to their number of base pairs, so this will be their size. The fragments that have less base pairs will actually travel further, while those with larger base pairs won't travel as far. And the middle well was used as a standard. That way we could actually compare. So we know for a fact that these are how many base pairs at each strand. And so we would then be able to compare our strands produced by the gel electrophoresis in order to determine how many base pairs were present. It's also important to know that our goal for this was to have about 550 base pairs, or a fragment of 550 base pairs. And the reason why is because we wanted to ensure that we had enough PCR product. And so again, like I just said, the purpose of this was to determine if our PCR was successful, and the fragments helped us determine the molecular weight of the DNA that was amplified, as well as a successful PCR PCR would provide us with 550 base pairs. Unfortunately, our PCR was not successful, as you should be able to see in one of the following images, as there should be a positive control with bands in a well and a negative control with no bands, and then there should be our experimental values, and our experimental values, as well as our positive and negative control, did not show up. So that actually means it's most likely that there was an error in preparing the samples for the gel electrophoresis, because at the bare minimum we should have seen a result from the positive control. So after doing that, we conducted a PCR cleanup, and the PCR cleanup was done with an Ampure XP kit, and it was manufactured by Agent Corp Incorporated. And the procedure was followed according to the kit, with no observations made, meaning we did not noticeably make any changes intentionally. And the purpose of conducting a PCR cleanup is to remove molecules that might have contaminated or have been left over from our indexing. And so when I say contaminated molecules I'm referring to are unused primers or excess components from the buffer, which includes enzymes or leftover NTPs, which refers to unpaired base pairs. So our methods when conducting or indexing PCR, we were adding a unique set of sequencing tags to the amplicon so that we may identify and analyze these samples later. So as a class, we were all going to be putting in different samples from different species, and we wanted to be able to identify our samples alone. And so this was done during the PCR reaction with the 3' which will attach to the amplicon, while the 5' will then attach a sequencing tag. Steps for this included holding at 95 degrees Celsius for 3 minutes, and then we conducted 8 cycles in which the products were held at 95 degrees Celsius for 30 seconds, 55 degrees Celsius for 30 seconds, and then 72 degrees Celsius for 30 seconds. From there, the third and final hold would be at 72 degrees Celsius for 5 minutes until it was stored at 4 degrees Celsius. After indexing the PCR again, we wanted to clean it up one more time, which followed, again, the same procedure as the first PCR cleanup. We used the same Ampure XP kit, which was, again, manufactured by Agent Corp. And, again, we followed the procedure according to the kit, with no intentional changes made. And, again, the purpose of doing this PCR cleanup is to remove excess molecules that may contaminate or initially just cause error in our samples. And so the next step was then to quantify our PCR products. And in order to do this, we used a qubit fluorimeter. And these work by attaching like a fluorescent dye to DNA molecules. And these fluorescent dyes will help us trace specific shifts in the fluorescence in order to determine, again, the concentration of products. And so the reason we use the qubit fluorimeter and with the fluorometric assay instead of the spectrophotometer, which again is what we used earlier to determine our concentration, was because it's more precise and it can distinguish between DNA and contaminants. It can also read lower concentrations of DNA and RNA. So the third and final major step of this was sequencing, and the instrument we used was the MISSEQ automated DNA sequencer. This was manufactured by the Illumina Corporation. And the purpose of sequencing all along, of course, was sequencing the various amplicons and of the scat samples, which allowed us to determine the various forms of bacteria present in the gut microbiome of our two species. And so you should be able to see here our UV spectrophotometer results. And if you're looking at these, you should be able to see that almost all of them, except for the 429 control, had a high enough concentration of our results. We wanted a 2.5 concentration. Unfortunately, for our 260 to 280 ratio, there was also our 429 original experimental value, 142, was the 260 to 280 ratio, meaning that this product might have had some contamination from a protein molecule. Here you should be able to see our gel electrophoresis results. Unfortunately for our samples, our PCR did not work. As you can see that there is no bands in any of our wells. That's really unfortunate. As I mentioned earlier, the result of this must have been error in preparing the PCR, because at the very least, even if our experimental values were not properly prepared or quantified, the positive control provided should have had bands show up in the well. So moving on to our discussion of our amplicon PCR. As I just mentioned, we were not successful in extracting the DNA from the feces. So basically the lanes in our gel did not show any PCR products, so there was not a high enough concentration for us to run or sequence them. This ultimately resulted in us having to move on and analyze samples of other species. So as we move on past this, we'll be talking about two different dogs that we analyzed, and we'll be comparing the gut microbiome too. So as mentioned, these are two different species. Again, our bearded dragon and snake are not successful, so this is a comparison of the bacteria found in the gut microbiome of two different dogs, and their graphs should be shown here. And so from those results, we were able to see that the two samples, one had 10,313 reads, and another one had 105,507 reads. And basically what this helps us determine for our kingdom species level was that organisms that were identified were 96.5% bacteria and a 3.5% other, with none of them were unclassified. And so there's an insignificant amount of unclassified organisms identified in our reads, which you can see because there is no representation on the graph. So moving on, we are able to determine that subject 333 had about 14,406 successful total reads, while subject 520 had about 165,000 successful total reads. And these numbers were essentially big enough for us to analyze. So the differences between the gut microbiomes of subject 333 and 520 were that at the kingdom level, the two subjects were pretty much the same, and identical at this level, both mainly consisting of just bacteria. And this is what we expect, considering they should both be dogs, so they're under the same key. So moving down the list from kingdom level to order classification, that's where we start to see a difference between the two gut microbiomes between our two test subjects. Their microbiomes for 520 show a lower percentage of 4 different bacterias, whereas subject 333 had a higher concentration or a higher percentage of those 4 bacteria present in its microbiome. So moving on to like a genus analysis of the gut microbiomes of our 2 species, we see a lot of different bacteria present in each species' gut microbiomes. For example, subject 333 has about a 60% presence of bacteroids, while subject 520 only has about a 20% presence of bacteroids. Also, subject 520 has about a 40% presence of the protobella, while subject 333 has less than 10% of a presence of the protobella. Moving on, subject 333 also shows lower levels, less than 10% of the 3 bacteria known as Blatia, Ruminococcus, and Alcophilus, while subject 520 has no presence of either of those bacteria shown. On the contrary, subject 520 shows low levels of Megamonias, Fusobacterium, and Salmonella, while subject 333 shows no presence of those materials. So overall, I guess what we can conclude from these results is that there is an overall difference in the types of microbiomes for species, especially if they're eating something different. This could relate back to our own specific species, the bearded dragon and the snake, because obviously if 2 dogs who are in close relation, I believe one was Yorkie and one was a poodle, if those 2 have a big difference or variation in the bacteria present in their microbiome, and they're eating similar diets, or what we hope to be similar diets, then it should be assumed that, a snake and bearded dragon should have a larger or a bigger difference in the bacteria present in their microbiome.